Welcome to today's lecture. In this video, I will discuss about the single beam UV visible spectrophotometer and double beam UV visible spectrophotometer. In single beam UV visible spectrophotometer, components are radiation source, monochromators, sample cell, detectors, galvanometer and readout devices. Now see one by one. Radiation source. Radiation source or in UV, hydrogen discharge lamp, deuterium discharge lamp, mercury arc, xenon discharge lamps are used. In visible, only tungsten lamps are used. Monochromators. Monochromators are converting the polychromatic light into monochromatic light. Conversion of polychromatic light into monochromatic light is the function of the monochromators. And in these monochromators, when radiation source fall on entrance slit will pass only polychromatic light through it. After reaching the collimatic mirror makes parallel the radiations coming from the entrance slit, makes the parallel radiations coming from the entrance slit. After the rays will fall on prism or gratings. It will disperse the light according to the component wavelength. It disperse the light according to the component wavelength. And after that, it will be reach on focusing mirror. Focusing mirror reforms the image of the entrance slit. It reforms the image of the entrance slit. After that, exit slit will select one spectrum for observation by the detector. And the next one is the sample cell. Sample cell. Only one sample cell will be there. In this, only one sample cell will be there. It is not possible to compare blank and sample solution at a time. Why? Because in this instrument have only one sample cell. This instrument is not possible to compare blank and sample solution at a time. Why? Because in this instrument have only one sample cell. After that, after that detectors, the detector will convert the light energy into electrical energy. Detectors will convert the light energy into electrical signals. When radiation source fall on sample cell or reference cell, some amount of energy, some amount of light will be absorbed through the sample cell. The remaining get transmitted and displayed on readout devices. The transmitted light get fall on the detector and it give the intensity of the radiation absorbed by the sample. And next, the galvanometer. It measures the output. Galvanometer measures the output and readout device displayed the spectrum. In this single beam UV visible spectrophotometer, the absorbance readings of both the standard and unknown solutions are recorded after adjusting the instrument to 100% transmittance with a blank solution each time the wavelength is changed. First, first the blank solution is kept in this instrument. After that, sample cell. After that, blank solution after that sample like this, uh, doing this doing the experiment in this instrument. And the next one is the double beam UV visible spectrophotometer. Components are radiation source, monochromators, chopper, sample cells and reference cells detector readout devices. In this also radiation source, this radiation source in UV hydrogen discharge lamp, deuterium discharge lamp, mercury arc, xenon discharge lamps are used. In visible only tungsten. Tungsten lamps are used. Monochromators are convert the polychromatic light into monochromatic light. Same in double beam UV visible spectrophotometer. Monochromators Monochromators when radiation source fall on entrance slit only passed through it, only polychromatic light only passed through it. After that it enters collimatic mirror makes parallel the radiation coming from the entrance slit. After that gratings or prisms will disperse the light according to the component wavelength, and after that focusing mirror will reform the image of the entrance slit. um, and next exit slit will select one spectrum for observation by the detector in these monochromators. In this instrument the monochromatic light is split by a rapid rotating beam chopper into two beams or two lights. One is for sample cell, another one is reference cell. And after that light, the remaining transmitted light fall on the detectors, and mm, detectors will convert the pul- convert the light energy into electrical signals, and it give the signals to the readout devices, and readout devices will display the spectrum. In this double beam UV visible spectroscopy, at a time we will measure the sample and blank solutions. At a time we will measure in this instrument. If you like this video, please like, subscribe my channel.